Ladies and gentlemen, thank you for still being here and listening to me. I'm going to present you today my research about the embryonic origin of the mucosal glial cells in the large intestine, and we carried out this research with the help of embryo surgery. But let's start from the beginning. So, what is the gastrointestinal tract? This is our organism that helps us process all the food that we ate. And here on the right-hand side, you can see a schematic picture about how it's structured. You can see, So in the center, you can see the lumen, and then you can see the mucosal layer, the smooth muscle layers, and in between, you can see the mucosal and the mesenteric plexus, which is part of the enteric nervous system. But what's the enteric nervous system? So the enteric nervous system contains the glial and neuronal elements that are responsible for maintaining the healthy gastrointestinal tract. It's responsible for proper motility, blood flow, absorption, secretion, and immune functions, as well as the microbiome composition. If, because of some development disorders, the enteric nervous system won't be formed, it's not going to be able to have the properties that a healthy gastrointestinal tract should be. Our laboratory focuses on Hirschsprung disease. This is a congenital aganglionic megacolon. In this state, the neural elements are missing from the colon, and because of this, there will be a spasm at this area where there's no neurons, and before this area, there will be the megacolon, which is basically a swollen part of the gastrointestinal tract because the spasm won't let the secreted materials to flow forward. And because of this, usually vomiting and distension occurs, and the occurrence of this disease is 1 in every 5,000 newborns. And the goal of my research is to find a cure for Hirschsprung disease with the help of stem cell therapy, because now the only option that we have is a surgical method how we cure this disease. But when we dissect the part that's not normally formed, usually the ones that we leave in the newborn baby still lacks the proper functions. The enteric nervous system is created by neural crest-derived stem cells. It's important to note that these stem cells colonize the colon from 2 different directions. First, the bigger neural crest cells colonize the colon from the sulca, and the sacroneural crest cells colonize the colon through the extrinsic innervation, and they are coming from the remic ganglion. In the corner, you can see different immunohistochemical staining that I used during my process, and the most important are the TUSH1, which is a beta-3 tubulin marker for neurons, and the SOX10, that's a glial marker, but also marks neural crest cells. Now that you know every basic aspect, let me show you a very interesting video, if I can. But somehow my presentation is not looking how it should, so I'm just going to tell you what should be here. So you should see the cells that we were interested in. We found a very interesting TUSH1 and SOX10 double positive cell in the lamina propria layer of the colon, and these two immunohistochemical staining should not occur at the same cell. So this is why we started focusing on this specific cell. And we wanted to find the origin of this cell. Oh, okay. So, just one second. So you can see with red, you can see the cell nuclei, and with green, you can see the cells, how they are connecting with each other. And this is a very interesting 3D confocal microscopy imaging. To find out the origin of our double positive cell, we created a choriantaloid membrane transplantation model. I used 5-day-old chicken embryos. I dissected the hindgut, and then I put the hindgut onto the choriantaloid membrane of a 8-day-old chicken embryo. The choriantaloid membrane is a very good place to further culture our grafts because it's widely vascularized and it can provide the proper nutrients for our grafts. There were 4 different setups. First, I removed the cecum and the remac ganglion as well, leaving the graft with 0 intervention. Then I left both the remac and the cecum on my graft, which should be the positive control of my research. This should represent the healthy state. Then I only left the cecum, and in the force I only left the remac ganglion, which should show us what happens if we only leave like the option A or option B in the graft. So as you can see, in the negative control you can find 0 neural elements, and in the positive control you can see a fully formed intestinal wall with the double plexus, so the submucosal and the mesenteric plexus. And the interesting part comes at experiment 4, when I only left the remac ganglion on my on the intestinal walls. And my research shows that with HU we would stain neural elements, but there were 0 neural elements, whereas with the GFAP you can see the glial elements, and here you can see the double positive cells that we were looking for. So this occurred when I removed the cecum, and now I have 0 idea what happened. Okay. So yeah, with the double immunohistochemical staining, yeah, we found this one when I removed the cecum and left the remac ganglion, which shows that these double positive intramucosal cells are coming from the extrinsic intervention of the colon. For further experiments, we designed a further in vitro culturing with GDNF, which is a glial growth factor. The reason we want to do it because glial cells have the capacity to transform into neurons, and we want to test if we can transform the intramucosal glial cells that we found into neurons as a possible therapeutic treatment for Hirschsprung disease that I spoke about in the beginning of my presentation. And the other thing is a transgenic mouse model that we want to use to validate our findings in chicken embryos to mammalian models as well. We used either NRB knockout mouses, and what you should see here is that on the left-hand side at C, you can see the part that has the spasm, and before that at B, you can see the megacolon. And basically at B, with HU, we stained the neural elements, but in comparison at section C, you can't see any neural elements, which also validate our finding in the chicken embryos. And furthermore, we used a WNT1 TD2 EDNRB knockout mouse. Here, if you look at the red staining, that stains all the neurocrest cells, whereas the green stains the glial elements, and in the right corner below you can see the double positivity as well. So as a conclusion, we discovered the network of intramucosal glial cells with the help of embryo manipulation. These cells express neuron-specific beta-3 tubulin TUJ1 and enteroglia-specific SOX10 protein. Intramucosal glial cells have not been located in the colon before. The cells are present in experimental aganglionic colon, becoming target of a possible stem cell therapy. Transgenic mouse model supports the neurocrest origin. And I would like to thank my mentor, Professor Nándor Nagy, for his help, and my whole laboratory team, and obviously NTAA as well. And if you have any further questions, I'm going to be here the whole weekend, so feel free to come over.
Thank you for the nice presentation, and now please take your certificate. Zsanna Gecse.